perhaps the most unusual location I've spoken at. And that's saying something. And I'm looking forward to interacting with you folks. And I'm really amazed at this whole Mumbai Local initiative and what Junoon has pulled off. I've seen some of the websites and it's a privilege to be hosted on them. Since I'm supposed to talk about rain, I'm going to start you off with a neurological exam. Is there a collar mic by any chance?
No, they don't.
Okay, I will do something.
Do you want a stand?
Sorry?
Do you want a stand for your mic?
It would be nice, yeah. Because I need both my hands free for
Yes.
So, can everybody take one hand out and hold it real still like so? Okay. Now with the other hand, hold something weighted like your cell phone. Yeah. Hold them both as evenly and as level as possible. Cell phone, cell phone, cell phone, cell phone. Yeah. Now, take one finger and turn your phone off.
It is.
Better check mine's off. What fun if it rang in the middle of this? Okay. Now, science is not an individual activity. Okay. Almost anything is not an individual activity, but science in particular, one brain is not enough. The truth is above us all, and we are little puny little, human minds trying to address some of it. So it really works when you have fantastic researchers working with you. So I'd like to introduce my team of students and postdocs. Suranjana, Zeba, Leora, Vitika, Bhavna, and soon to join us, Priya. I have stooges in the audience. Science is also a real experimentation. I do neuroscience in the lab, and at home I do little neuroscience observations on two little brains that are growing under my care. So it's all brain work everywhere. And we're going to begin now with a little demo. So I'd like each of you to take those sheets that they're handing out. And by the end of today, you will have memorized its contents. Please do not read them, they're just papers. We're being eco-friendly, so we took papers that we know won't be useful for anything else. Okay, the content is not important, just that it's a paper. Give me one too. Okay. Roll it up so it's a long cylinder. So roll it on its long axis. And I'm going to talk without the mic for 2 minutes because I need both hands. Sorry, we rehearsed everything and we didn't rehearse this. Actually, can you hear me if I don't use the mic?
Yes.
And in terms of the video, is that okay? Yeah. Okay. Alright, so now, this is just an exercise. This is not about cell phones or anything. Actually, let's not have this bright light staring at me. Hold it like so and pretend it's a water tank and touch your palm to it like the level is high and now the level is low. Sorry. Yeah? High to low, touching your palm. Okay. Now you're going to do the same thing in front of your eyes. Keep both eyes open and keep this in front of your eye, one of them. And block the other eye with your palm, touching the cylinder. Okay, so you should ideally be seeing out of only one eye right now. What do you see? Look at me through the cylinder. What do you see? Okay, that did exactly work. I don't have the physique. Keep looking, keep looking, keep looking. And now. So your palm should not be squishing the eye shut. It should be just lightly over the eye. Both eyes are open. And now, the palm is touching the cylinder and it's sliding along the cylinder all the way to the other end. Keep it there, keep it there, keep it there. Keep it at the other end. You're not blocking the cylinder. You're looking with one eye through the cylinder. Yeah. What do you see?
Hole in the palm. A hole in the palm.
See a hole.
She said there's a hole in her palm.
Yes.
What if you had four? Okay, what do you see? Keep the palm right to the end of the cylinder and look with both eyes. What do you see? one eye looking through the cylinder, the other eye looking normally, but then your palm is there.
Cylinder is center, at the center of the palm.
The cylinder is in the center of the palm. How many see that? How many see something else? So this is how I begin. Yes? Me and the palm. So it's like this funny photographic effects, right? You're seeing. Okay, now you're seeing this, right? Seeing is believing. If you were put on a witness stand, you would say, I saw Shubha Tole through a hole in my hand.
That's right.
That's fact, right?
Exactly.
I mean, you know, seeing is believing, and eyewitness testimony puts people in jail. When I begin my neuroscience class, I ask the first years to debate, and I'm going to ask you to debate. How many of you would say the hole in the hand is real? And how many would say the hole in the hand is just something created in the brain? Real and created in the brain. How many go for real? One. Only one person thinks the hole in the hand is real. You saw it?
I saw it.
How many people think that the hole in the hand is something created in the brain?
I would call it illusion.
Illusion, okay. So one person thinks it's real and everybody else thinks it's an illusion, but you saw it.
That's because we're pure. Because we're pure and we're
You saw it because you're pure and you're with me. Now, how many of you think the chair and this thing and whatnot that you see is real? You see it, huh? Okay. How many of you think that the microphone is real and continuous object now?
Because we know it by experience. And now when we
Because we know it by experience, you say it's continuous.
Yes.
But what are your eyes telling you? Do you know you can do tests with infants, 3 months old, 6 months old, okay? And object completion can be tested. How do you ask an infant who's 3 months old? I mean, they don't even say that, right? There are lovely tests you can do. You put the infant in the mom's lap and you give the infant something to suck. And when infants are curious or interested or they're saying, no, that's weird, they suck faster. Okay? So you can test infant mathematics. Okay, if 2 balls go behind the screen and then you remove the screen and there's only one there, they go. You know? It's amazing, okay? And object completion can be done by infants, okay? These things are not all learned, because if they were all learned, you would not realize that the tiger with the stripes behind the trees is actually a tiger, and you would take your time learning what is visual illusion, what is not. Okay, so there are things that are wired in our brains, okay, to extract shapes from incomplete things. This is how our brains are wired up. And I submit to you that the hole in the hand is as real as anything else you see. Okay, I submit to you that everything you think is real is actually a construct in your heads. Because we poor, pathetic, puny little humans, so proud of our lovely brains and everything, we are completely limited by what we feed into those brains. And let me show you what we feed into those brains. So what we study, this is the law of the Tata Institute of Fundamental Research. You could block it, they can just imagine it, it's the same. So what we study at the Tata Institute of Fundamental Research is how to build a brain. And the reason we study how the brain is built, because how it is built actually shapes how it functions. Okay? If the wires were connected this way or that, it would actually change how your brain processes things. I talked about hole in hand and vision and so on, and we'll look at the visual system a lot because it's just interesting. You know, you can imagine things and see things. But it works equally well for other sensory modalities. Now, this represents the auditory modality, certainly music. It also represents the visual modality, see, black and white. It also represents the touch modality. Okay, now, you would not be able to see those black keys as different from each other. You would see them as one blur of black. Okay, if you didn't look at them with your fovea, the very center of your eye, of your retina. Okay, if you look at the side, you're less able to distinguish. So if you were asked how many keys are there, You would have different answers depending on which part of your retina you're looking at. Now tell me, which is real? Okay? Let's take sensation. You know, there are different, our bodies are not equally well, they don't have equal access to the brain. Okay? You think you're one person, but little bits of you are fighting for access to your little brain. And just like you would buy a larger and larger megapixel camera for better resolution, right? And a camera is faithful and true. It will give the whole field of view equal resolution. Your eye isn't doing that. It's saying, my goodness, that's not an effective way of looking at the world. It's cramming in the receptors into the very center fovea. And the reason you know what's out there is because you foveate over everything. Okay, you actually form a map of the world by foveating over it. Can you imagine the mechanisms that have to control that? And your sensation, okay? If you were to feel the piano with your wrists, you would not be able to feel two keys as separate keys. The body is very careful in how it uses its real estate here. It puts all the important sensitive areas that need discrimination, it gives them access to the largest amount of megapixels up there. Okay? So, neuroscientists in maybe in the spirit of fun, or maybe in the spirit of trying to represent how this actually happens, drew a picture of what a human would look like if you change the body parts in proportion to how much area they actually connect to in the brain. Okay? So which body parts would you expect to be big? Which body parts would have lots of access to the brain? Okay, before people start guessing embarrassing things, let me just show you. Okay, so this is the body surface in proportion to the cerebral cortex, the cortical area that it connects to. And here you go. This is you as your brain feels it. Okay, this is the feel map in the brain. You can see certain areas have access to a lot of real estate. So you could do a lot of fine discrimination. You could tell two grains of sand apart if they were on your tongue perhaps. Right? But some other areas, not really. Okay? So how you see the world is highly distorted based on how the sensations, you see the world with your senses, right? And how your brain distorts the senses is completely shaping your internal idea of what the world is. And this somehow, so being a neuroscientist I find allows very humbling feelings and sensations, right? Because you realize how nothing you actually think is true and hard and fast is true and hard and fast. Everything you think and feel is just limited and constrained and shaped by how the brain grew and how it's wired up. And in fact, our whole, so if this is true of senses, if you can see hole in hand and believe it, and if you can see that a mic or an arm between the slats of a chair is a continuous object, I mean, almost anything is possible, right? And if your sensory surface is shaped by how many receptors connect to how many parts of the brain, everything out there is molded. Okay, molded for what? Molded for, I would say, survival. Okay. The one thing a species does in order to continue is to reproduce and survive. So that is the one guiding principle, I would say, that shapes everything. And every species does it. We are not that different from a tiny little fish. I'm going to show you a little video. What will be the first 24 hours in the life of a fish. Okay. This is a fish egg. It hasn't waited for us. One cell has already become two. And very soon, as you watch, the cells will divide further and further. You'll have a ball of cells, and before your very eyes, a little fish embryo will appear. Watch and wonder. Right now it looks like nothing, but give it 2 more seconds. And suddenly you see an eye popping up here. While you're looking at the eye, look, the beginnings of the spinal cord have popped up there. Okay, it's a fish. In 24 hours, it became from a blob to a little fish that will actually twitch and move. I mean, what did you and I do in the last 24 hours? You know, nothing we do in our adult, mature lives, nothing we do in our childlike lives rivals what we do in early development. And in fact, a grandfather of developmental biology, Wolpert, said very, very accurately that it is neither birth nor marriage nor death, but this process that is the most important event in your life. Afterwards, it's all downhill. Okay. Now, what this creature does is to build its control system here, its brain. Its control system is not one block. Its control system is actually many specialized structures. Okay, you have the visual area, and it is not one area. As you will see later, the visual system is made of many, 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 many areas. You have the memory center, which is not just one area. It's not a place where memory is stored. It's a place where memories are made, and it has subparts. You have language, which has subparts. How are all these subparts made? So my colleagues and I have put together a little demo for you. So let's invite them. This actually represents work maybe farther up there. So we're going to show you, are we okay? We're going to show you a representation of work that we did at Tata Institute that figured out how you get complexity from simplicity. Okay, so what you see here is a row of very similar looking, undifferentiated, naive young brain stem cells. Okay, they're stem cells. They do what stem cells are supposed to do. They divide and make more of themselves. They create specialized cells. Not yet. Okay, right now they're just undifferentiated stem cells. And they are a uniform sheet, a very uniform looking sheet. And what the brain does is it's come up with a cool mechanism for creating complex structures from a very simple sheet by not making all the complex structures at once, because then you make mistakes, right? If you suddenly got to do 8 different things, you're going to get one or two of them wrong, and that's disaster, especially at this early stage. So what the brain does is it solves the problem by, instead of making 8 different things at once, it just makes 2 different structures. It divides this complex sheet into naive undifferentiated stem cells, and from one end of the sheet, it turns off a critical gene. So stem cells at one end of the sheet don't have this protein anymore. Let's take it away. Oh my God, she doesn't have her special protein, so she's no longer a stem cell. Oh my goodness, poor thing. Now, sometimes losing a gene, losing a protein is in fact as effective or more as gaining something. We're all built on this positivity, right? You have to have a gene, you have to give. Well, we've taken away one particular gene, okay, that my lab discovered is important for this process. We've turned this one cell or this one group of cells into no longer cortical stem cells, but into something else. Who are you, Zeba? She's a signaling center. She's a powerful, radiant, magical signaling center, and she's chucking signals across at the other stem cells. Okay, this actually happens. Okay, this actually happens. And for reasons of strength of reach, okay, the stem cells nearby get more signal than the stem cells farther out. And this is how you create 1, 2, 3, 4 different kinds of structures out of a simple sheet. So now you have differentiated stem cells. No, they still have the gene that makes them cortical, because if you lost that gene, you became the signaling center, the lighthouse instead. So the brain uses a clever mechanism of making a lighthouse and making responsive cells. And these responsive cells respond differently because they got different bits of confetti thrown on them. Okay, so this actually happens in the brain, and now you've created a complex structure. The structure that we explored a little bit is the hippocampus. It processes learning and memory. Now, because these different bits of the hippocampus already know they're different, they now extend connections in a very specific, directed way. And they're not that simple. Hippocampal connections are not so simple, sometimes they're complex. Because the hippocampus is actually the recorder of memories. It's here that you record a memory and then send it off someplace to be built. Now, what happened to our lighthouse here? Okay, completely useless and done away with? Well, it shrivels away with age, but before it does that, it actually serves as a nice site to attract nerves from the hippocampus. Okay, they actually all funnel this way and then go out to do what hippocampal nerves do. So this is sort of a little skit that shows how brain cells sort themselves out and turn a simple sheet of cells into something completely different. So we ask our happy hippocampus to now innovate itself. So we showed you now how brain structures are formed, how they are specialized. Okay? So imagine now all over the brain. Indeed, I showed you hippocampus, that was our work. But others have shown that similar signaling centers are created elsewhere that actually tell your sensory, so everybody, all of us, and that's the brain, not just all humans, but all primates, every bandar you've seen on your way to Mathiran, every mouse on the street, every mammal reproducibly has vision, sensation, motor, auditory, okay, and hippocampus buried in the middle. This map is invariant. It's so invariant that even though the mouse has a cortex about the size of a 1 rupee coin, and the monkey has a cortex the size of a pulki roti, and we have a cortex of a large size pizza, this map is invariant. You do not see a mouse with the visual cortex up here, because if that mistake happens, enough has gone wrong with how the signaling center formed that many, many other mistakes happen, and that embryo would not ever get born. The development is controlled very, very tightly. And why is it controlled tightly? Because the job is complex. Okay, we had far, far too many neurons to make mistakes with at this early level. So this, for example, is a nice color-coded view of a bit of cerebral cortex. It's like a millimeter of cerebral cortex. Cortex is a sheet, and it has 6 layers of neurons. So they are color-coded, and you can see these yellow lines. So these are the input wires of a neuron whose cell body is here, and the output wire comes out this way. So you can imagine how complex it is, and yet how remarkably well ordered it is. Because all of these neurons were born from cortical stem cells that received signals to create just the right type of neuron. And creating the right type of neuron means telling it which way to grow its wire. Up, down, sideways, whom to connect to. Because finally what the brain has to do is function. And if it's not wired up correctly, it's not going to function correctly. By the way, the hole in the hand was evidence that your visual system was wired up correctly. Okay, the visual system is supposed to take 2 slightly non-matching views of the world and join them up. That itself is weird, okay? I continue to hark back to philosophy. Okay, there's 2 views of the world, right? They're not exactly the same. Why shouldn't there be 2 bookstores out there? Why shouldn't there be, a double pillar out there? Why is there one? Somebody say, please, I walked into it and I felt it was one. But then you're using your sensory modality to corroborate what your visual system told you. So we're living in a matrix-like world, as it were. So, neuroscientists watch these films with great, oh, somebody's really saying what's real. Actually, forget Matrix and forget Hollywood. The ancient Hindu sages had it right, okay? The world is Maya. It's actually all in here, it's not out there. And it's so fascinating that we're spending our lives studying it. That past Matrix. So what's one neuron then? Let's simplify this. Here's one neuron. One simple neuron. Okay, this thing is nothing but simple. Okay, this is not a model system for anything. A model system is a simpler version of what you want to study. This is complex. What's each little red dot? Each little red dot is where some other neuron has connected to it. Okay? There are lots of connections. The brain has about 100 billion neurons. This number is thrown about. And each makes 1,000 to 10,000 connections. Each one. Okay. Before they make connections, neurons actually move to their right. They're the place they're supposed to be. And I'll show you a little video that I borrowed from a colleague, Kazunori Nakajima, from Japan. And just enjoy. These are these red things. Each of them is a newly born neuron that, because after being born, has sort of realized what type of neuron it is because it received the right signals. And now it's going to wander off to its destination. I mean, they know what they're doing, okay? Even though some of them go back and forth a bit. Want to watch it again? They really do seem to know what they're doing. And at the end of all this, they get wired up, and then we have a brain. And then we see a hole in the hand. Okay? Here is another video, one neuron in a dish. Okay, just to give you a sense of how complicated it is for even that one neuron to grow. I have a hyperlink here with that. ball in a dish. It's playing on my screen. This is playing there. Should I ask one of my students to come and do the dance of the growing neurons? We're nothing but not enthused about science communication. Is the video somewhere we are doing, or shall we just go past? Alright, you will not see a neuron growing in a dish. We will leave that as a suspenseful thing. Alright, this then is a view of the brain, and superimposed on this brain, I've put a whole bunch of structures. We just told you how the structures were made, we said they grew wires, and this is your visual system. This is a simplified map of your visual system. The complicated map looks worse than maybe a few hundred underground maps of the London metro system all piled up one on top of the other. Okay, this simplified version is how we see. Okay, and we're going to sort of bring out how strange it is that we see anything at all, because on the one hand there's a lot of information out there. On the other hand, it's being taken in through these very, very limited organs. There's only so much type of signal they can encode, and out of that we construct the world. I mean, who knows what's actually out there? Okay, I'll give you a little example. We see in a certain spectrum, right? Visual spectrum. If you were to see in ultraviolet, the world would look very, very different. You wouldn't see all these colors at all. Okay, now, what you see, okay, is dependent on, or the size of the smallest object you see is dependent on the wavelength you use to see it. So, that's why when you want to see something really, really small, okay, you go to something called the electron microscope. When you want to see something even smaller, you use X-rays, okay? Because you can sort of imagine, if the only light I had to see you with had a wavelength of like a meter long, I wouldn't be able to see you two apart. Okay, you'd be one thing. So what we see is completely limited by the wavelength we're using to look. If we could have Superman's X-ray vision, okay, Superman actually, if he really had X-ray vision, he couldn't function in the world. Because he'd see the atoms and molecules of everything out there. How would he be able to move? He'd be able to walk, everything would, yeah? And if you look at a piece of wood, it's not a piece of wood, it's got holes, because you can see all the space between the atoms. Okay? So, I wish you to leave today, I'm not wrapped up yet, I'm just halfway done, but leave today thinking about what's really out there. Is it out there or is it inside? And these are the delights that continue to confound us, even as we do experiments with, neurons and brains and so on. All right, that is the simplified visual system, and I'm sure you're a little bit terrified as to what I'm going to do with next. So I'm going to pose this question. जब डेज टाइम बॉलिंग करते हैं, विराट कोहली के दिमाग में क्या चलता है? Okay, I assume at least 90% of you are total cricket fanatics. Can I ask the ones who don't know much about cricket to identify themselves? Nothing bad will happen. I'm not there. So, in fact, to come up with this title, I have to ask my own resident cricket expert here. Who bowled well recently and who batted well recently? Because the one thing that a cricket batsman has to do is to have good sharp reflexes. You have to face projectiles coming at them. And this is a very strange skill, right? Because usually when a projectile would come at you, what is your instinct? Yeah. That would be the safe thing to do. That's what nature and evolution, billions of years of evolution taught creatures to do. When there's a projectile coming at you, run, get out of there. But not our heroes. Sorry. They don't run when something is coming at them bowled by Dale Stein at 150 kilometers per hour because their life is not as precious as those 3 sticks behind them. Okay, brain can learn. It can learn to override its evolutionary programming. Okay? For this, the brain has to have real quick instincts. And you know, we have so many brains in the audience. When a neuroscientist looks at an audience, they don't see an audience, they see brain. We're going to test your instincts. Let's go. We have here some of the best minds of the Tata Institute of Fundamental Research. Go ahead and test their instincts. Please do go and throw them all back, okay? Take them as souvenirs, our next outreach won't happen. Almost. Next. Okay, who fought and who ducked? Who fought and who ducked? Confess, nothing bad will happen, they're all okay. Who fought? Right. 1, 2, 3. Right. Who touched it? Who ducked? Clever. How do you know what I'm throwing at you?
I didn't do it wrong.
You didn't even. Fine. You read Zen like Nirvana. What's out there? What's inside?
I didn't do it, nothing came to me.
You observe these crazy people.
Yes.
Okay, so catching and ducking are just two variations of a decision that's executed by your nervous system. Okay? I mean, you know, when a batsman's facing this 150 kilometers per hour ball and deciding what to do, it's a decision, right? You have to execute it. In fact, while you might try and execute a particularly good stroke, much of the motor decisions that we take on a daily basis are in fact not conscious decisions. They are decisions that our system just makes. Okay? We don't even know it. So we're going to involve you in a little activity. Nothing will be thrown at you anymore, I promise. To map out what happens when you see a ball being thrown at you. So we're going to ask various members of the audience to volunteer themselves selflessly in the cause of science and give us a few neurons from their heads. Or little neurons. As we tell you, if you would look under your chairs and find placards on the front row only, not everybody.
Only the front.
Keep some surprises. And if you could wear them, so, not yet. So, if you can stand up and do a sort of pirouette and show people who you are, your neurons of the eye. And there's 4 of you to represent the 4 types of neurons we see in the eye. Okay, red, blue and green sensitive and black and white sensitive. So now we're going to give you wires. So now you're going to extend your wires into the brain. Now there's many centers in the brain, but we've got under placards over there, seeing brain, the first stage of the brain, where the red, blue, green and black wires will extend in a painful map. So you can wear them and show people what you have. Now, what would happen if the red, blue, green and black mixed at this stage? The signal would get mixed. If it's mixed, then you can't ever separate it out again. Okay? So red, blue, green and black. Now, poor old black and white channel, okay? They don't process color. But they're special. The black and white channel is the fast channel. Think, if a projectile is coming at you, okay, you don't really care what color it is. You don't care whether it's got texture, what shape it is. The damn thing is coming and you have to compute how fast it's moving. So the black and white channel is a fast channel. The detail, the texture, the color, all that is a separate channel. And they're indeed called the what and the where pathways in the brain. So we have the where pathway holding a black wire. Blue, okay, blue, green, red, black. The where pathway. So connect the eye to the brain first. You can give your corresponding. This is only a temporary connection, don't worry. And you can wrap the wire around your wrist and you make it a nice tight wire. And then the seeing brain will connect to the what and the where areas, which are actually very different, okay? They're quite far apart in the brain. Because these things are processed very differently. So, yeah, you can go to where's what and where.
This might be fun if you went up and get them up.
It's the information, you've lost that, right? If you mix, if two red and blue neurons fire me, then I'm on, then nobody above me can tell what color I got. So keeping it all mapped is important. Yeah? Yeah. Okay, well when the circuit is all mapped up, we get you to raise your hand and show the big spider web. Okay, what was that? The where area is going somewhere. Wait, not yet. I have this area. Now what do we do with all of this information? Okay, we have to process it and take a decision on what to do. So we've got an area that's a little ahead of the motor area, the premotor area, which I've called decision. Okay, we don't, now here we represent just one gentleman with decision, but we have more than one decision neuron in the head, okay? Otherwise when that guy goes, no decision. And now, funneling into decision comes all of your training. This is what keeps a batsman at the crease, training. If I run away, what will they do to me later? Okay, so that training comes in as a memory of everything you learn. And since I said we have no idea where memories in the brain, we just put them randomly. Okay, they happen to be next to each other. If you don't mind standing somewhere else, you look more interesting. So memory just is stored somewhere. Okay, it's recorded in the hippocampus but stored in unknown places in the brain. That's why even if you lose your memory recorder, you still have your old memories that were recorded before. Okay? So there's memory coming in, nice yellow wires, all funneling into decision. And now what? This is a decision. What do you decide to do? Square drive. Square cut. Are there multiple copies of memory? I don't know. No. If you remove the recorder, you still have your stored memory. Okay. How they are stored? Well, probably the strength of connections of neurons and patterns. But copies, nobody knows how it's stored, so we cannot tell whether there's copies or not. It might be a good idea to have copies, but I do know that you can elicit a memory by connecting it to another memory. So, I mean, we all like, what's the name of this person? What's the name of this? It sounded like a flower or something. Okay, so you can trigger it. I don't know about multiple copies. Okay, I know that memory is a very, very strange thing. When you put something in long-term memory, it's there. But when you recall it, that process of recalling the memory makes it malleable. Okay? And when you recall it, it changes a little bit, and then you re-put it into long-term memory. So there are things you remember about your childhood well, because you have recalled them many, many times. But I can tell you that the way you recalled it now is not the way you originally recalled it the first time around. It's changed a little bit each time it goes back in. Okay? So if who we are is answered by, we are a sum of all our memories and experiences. If you recall them too often. Okay, we keep going back to this because this is what really inspires me as a neuroscientist. What is it? Who are we? What are we made of? Okay, and it seems to be like, the mental image I have is we're going down a slope on a little plank of wood, okay, catching rolling marbles. So you catch this marble and that marble and you're rolling along. And who you are is defined by the marbles you happen to be rolling on. And then when you lose your marbles, yeah? Okay, so now we have a decision. And the decision, whatever that be, is communicated to the motor cortex, the movement area. Okay, we've got, I think, one movement neuron who's going to execute. So, decision, by the way, if you inject a little current into a decision neuron, shall we try that? Okay, the person will move whatever it is that neuron was encoding, okay? And the person will say, oh yeah, I decided to move it because I wanted to. Alright? This also impinges on the whole discussion of free will. Okay, ideally, the movement neuron, when the movement neuron fires, the person should say, okay, I made a decision, I know I made that decision, and now I've ordered my movement neuron to fire. You know what? The consciousness of thinking you have now made a decision comes a little after you've already activated this circuit. So which means you think you've made a decision after some little person in your head has already made a decision. So there goes your whole thing about free will. In fact, I read studies that show that you can negate a decision, okay? Much sort of, if you have more negative control on a decision than you had in executing it in the first place. So instead of free will, there is free won't. Okay, so I'm throwing out all these terms in the hope that some of you will Google them and take yourself down this sort of rabbit hole, Alice in Wonderland like situation of what is it that we really have in our minds and what's out there and what do we make of the world. So we have movement and that's still in the head, but still in the head. Finally, who has to move? Arms and legs, no? Finally, move. So you connect then to the spinal cord via the cortical spinal pathway. We have spinal cord somewhere. And we have spinal cord connecting to arms and legs. So we have 2 arms and 2 legs. I think you guys have legs. Our legs right now. I should have put memory up there somewhere.
Where exactly is memory?
Nobody knows where it is stored. It's recorded by the hippocampus, but about the most famous patient whose situation allows us to understand this, known as HM by his initials. He's no more, so his full name is now known, so you can Google it. But HM had severe epilepsy, severe seizures, and because of which he underwent a bilateral hippocampectomy. Doctors removed both sides of his both hippocampi. Okay, they didn't ever do that again after that, but they did for him because his epilepsy was so severe. And he lived his life in a sanatorium. My postdoc mentor met him, as did many researchers who were trying to assess his ability. And it's very interesting, she said, if you meet him and converse with him, he sounds like a perfectly intelligent person. You can have a conversation. How are you? What is today? This and that. But if you left the room and came back, you would have no idea he met you. Okay, so he has a shifting time window of a few minutes. Okay, that's the memory that you can retain, but he couldn't store it because he couldn't record new memories. Okay, it was all shifting time window. But if they showed him a picture of himself, himself current, older, he would say that's my father because every memory up until the operation he remembered. So he remembered the World War, so memories that he formed before and stored. So this is proof that the hippocampus doesn't hold the memories because the memories were made there and stored elsewhere. And nobody knows where memories are stored because you can recall memories even as your hard disk starts degrading. Okay, you have dementia where bits and pieces of the hard disk start degrading and you can sort of still recall it after, you connect to this and that, you make mnemonics for yourself and then you do lose it altogether, unfortunately. But so that's the evidence that the hippocampus is a recorder, not the storage site. Yes?
One question. These connections between parts of the brain,
Yes.
are these permanent connections or they can vary and change?
Are the connections permanent? Are they permanent? Can they vary and change? What if they grow haywire? These basic connections are hardwired. Your visual area isn't going to suddenly, your retina is not going to suddenly start talking to motor cortex or something. But they are infinitely plastic. So if you don't have visual experience for a long time, if you don't, or if for reasons of accident or experiment, okay, 2 of your fingers are stuck together and they continuously experience the same sensation, the area representing the 2 fingers as separate becomes merged. Okay, so you lose resolution if you continuously stimulate the same area together. So, I don't do these experiments, so I think the better answers are found on the web. But I can just give you a flavor of what's out there. In songbirds, songbirds rely on their own hearing to verify that they're singing what they're singing. So if you play them a slightly time delayed version of their own song becomes haywire because they're trying to continuously. So there is a lot of plasticity. Blind people who have not much usage of their visual cortex, it turns out that the sensory cortex next door takes over. And when they read braille, actually visual areas get affected. So there are fantastic studies that demonstrate that the brain is not as hardwired as we thought it to be. Okay? But the basic map has to be there. You cannot suddenly have things randomly. Things tend to not just randomly grow haywire, they're usually logical. And I would recommend for you a marvelous book by Ramachandran called The Phantoms in the Brain. Okay, it's a popular science book, Phantoms in the Brain. it talks about the phantom limb phenomenon. When you have an amputation, you can actually think you have your limb, and then when you touch this with your face, you think your limb is being scratched. Fantastic book. But if we can just sort of complete the circuit, we have arms and legs now, and now I think we have a full circuit from having seen a ball to arms and legs. So shall we test this circuit? Okay. First, shall we have a photograph of the whole circuit? If you don't mind raising your hands, and if we can request our photographer to, yeah. I don't know whose brain this is. It looks like a giant spider. Giant spider web brain. Okay, and now let's test the circuit. So what I'm going to do, you can keep your hands down. I'm going to ask the circuit to fire in sequence, okay, by clapping. So we're going to go I as 1, C break is 2, what and where is 3, decision is 4, movement is 5, spinal cord is 6, arms and legs is 7, and memory can fire whenever you like because we have no idea when to do. So I'm going to go 1, 2, 3, 4, 5, 6, 7, and let's have you guys clap. So that's sort of like the signal going, okay? Ready, go. 1, 2, 3, 4, 5. Come on, you're bored already. Okay, we're going to do it at this speed. 1, 2, 3, 4, 5, 6, 7. Okay, ready, go. 1, 2, 3, 4, 5, 6. Okay, thank you very much. This was a good brain activity. I'm going to now move to an interesting animal in terms of execution of movements. Okay, an animal more interesting than Virat Kohli. Is that possible? So, okay, you can undo your ribbons, and I will just set up the PowerPoint again to.
Can I ask a question?
Yes.
Saying all this,
Some people express themselves in amazingly creative ways. All of these, I would say each one's circuits hum in different modalities, some better, some worse. So,
there is reinforced by brain.
So, I'm trying to say that each of us are set in some slabs of things that we can achieve based on our circuits, the genes, the environment that shape those genes, and then within that you can train. Okay, so sometimes these slabs are pretty limiting. For example, to be a fast sprinter, to do the 100-meter dash, you have to have a particular type of fast twitch fiber. You cannot train to be a 100-meter dash. Okay, to run a long marathon, at Olympic levels, you have to have a particular type of muscle fiber that doesn't fatigue very easily. So there are limits within which you can train. So depending on how you want to pose the problem, yes, there are limits, and yes, you can push and achieve what you never dreamed possible. And there are individual differences which we should actually celebrate. I mean, we are not all uniform, which is thank goodness, right? I mean, I enjoy studying the brain, but thank goodness somebody went and invented the cell phone. Yes. Thank goodness or not.
There is one question.
Yes.
They say human being has an unlimited, capability. Only the brain limits it. If you believe it, you can do it. How true?
Human being has an unlimited capacity. Only the brain limits it. If you believe it, you can do it. I would agree with you and disagree with you. Again, it depends on how you want to frame the question, right?
If I am limited by this room, and if I think that I'm not safe if I go outside this room, my brain will limit me from going outside.
I completely agree. So you can overcome
similarly,
fear.
we can extend this logic.
You can overcome fear. You can overcome many things. You can sing better than you ever dreamed you could because you thought somebody told you could never sing before and you didn't want to, but then you try and suddenly you can sing much better than you imagined. However, no matter how much you try, you're going to be limited by that wall and you can't walk through it. So depending on what you're trying to say.
To what extent this is?
Yeah, I mean, you can't jump out a window and start flying, whether you hold a feather in your nose or, a cape. So, I mean, obviously, right? So, certainly, the power of all positive thinking is going to shape. I mean, it shapes how medicines affect you. Okay, there are placebo trials and so on, where if you think you've been given a placebo, the medicine doesn't work as well. So, certainly, the circuits shape many, many things than we ever imagined. But
does it help in healing if you think about it?
Does it help in... So, I don't know, I haven't seen the studies, would be the scientific answer, but I would not be surprised if it did because your thoughts shape your immune system. And if your immune system is full of the right interleukins or the right cytokines, it's going to change how the moon heals. So if you're stressed, you're going to have the wrong things floating around in your skin's not going to heal. So that's just biology.
So healing can be expedited, right?
So the thing is, it's in the realm of science if you can address it by a scientific experiment. Right? So these things are tractable, some things are not yet tractable. So I would say science is not sort of, sometimes, I don't know if I'm actually addressing your question, but sometimes people try and use the word scientific when they want to communicate that something's important. I would submit that there are many, many things that are important that science is not yet able to address. So I would say science is this limited sphere, that which you can address in a controlled experiment. There are lots of important things out there that you don't, that science has not reached yet. So that's sort of, so the positive thinking thing, some of it has not been tested, but I would not be surprised if the brain is able to affect things much more than we ever had.
There is perhaps one more dimension beyond intellect, which perhaps science doesn't know, can't
Well, science doesn't know so many things.
Well,
yet,
intellect
as
itself
she
is
says.
such a loosely defined term. I mean, what is intellect? How quickly you can do math?
limited
I mean,
by
maybe you take 3 more seconds doing math than the next guy, but maybe you can appreciate a particular musical pattern quickly, which is intellect. I wouldn't go there. So we restrict in a very tight scientific discussion something that we can rigorously test. So let me go to the sort of last segment of this. I don't know how much time do we have. What is it? Okay, so this is like a 10-minute sequence, just to get you to see how fascinating is it that the brain, the job that the brain does, even when you're doing something as simple as running. Okay, so we're not going to go into metaphysics and the power of healing and these things that are not easily tractable. We're going to just look at motor functions running, and we're going to do this in an interesting creature. Not a batsman, not even a human. We're going to do this in a horse. Just look at this marvelous creature. You can also look at this creature on top of the creature, but right now I'm looking at the one who's running. I'd like to introduce this photographer scientist, Mybridge. Okay, this is an animated movie that has been put together from stills that Mybridge took at a time when high-speed photography was not existent. Okay, and Mybridge took these stills, actually in a farm near Stanford, I was told. He addressed a very interesting question. Okay, at the time, artists would paint pictures of scenery, animals, and horses galloping with their legs stretched out. This sort of the horse mane flying and the legs stretched out. So the question was, is there a time during a gallop that a horse is actually airborne? Is there? Or is it that they always have their feet on the ground? So Mybridge did the experiment. Made a horse run and actually set up tripwires and cameras and took time-lapse photography the hard way. Okay? And looking at this, it was very, very clear. Look, there is a moment when the horse is airborne. You can see it, right? That's science. This is incontrovertible. But he also, I think, embarrassed a whole bunch of artists. See when the horse is actually airborne? The horse is airborne when the legs are together, when they're stretched, right? So this is a story I heard. I don't know which parts of it are true, but the Mybridge certainly does have the credit for this. And this is an amazing species then, the horse, who is right now performing a gait called the gallop. If you look online, you will find that the horse can do about 5 or 6 natural gaits and another 5 or 6 more trained gaits. So this is what I mean by saying the brain can be trained and circuits can be trained. Now what is a gait? A gait is a sequence of steps that an animal takes to move. Okay? Now some are diagonal gaits and some are one side then the other side, and look. Okay, so this is a walk. Here a horse is walking. And can you try and see which leg it puts out when? Okay? I looked at it as carefully as I could and I find the sequence is 1, 4, 2, 3. Okay, the front 2 legs, the arms are 1 and 2 and the back is 3 and 4. It's doing a diagonal gait. It's doing 1 and then 4 and then 2 and then 3. So in fact, shall we have the arms and legs come out and do the walk for us? Look at this and figure it out while we come and ask them to. Okay. Okay, so we have the 2 arms be 1 and 2 and the 2 legs be 3 and 4. 1 and 2. You can face this way. And 3 and 4. You mind? You can have somebody else substitute for you. We don't mind. Okay. Now, the front legs and the back legs, okay? Arm and leg. Why don't you just clap instead of moving first, okay? Clap. 1, 4, 2, 3. Go. 1, 4, 2, 3. 2, 3, 1. Okay, why don't you now move with your clap? Take small step. Ready? Go. 1, 4, 2, 3, 1, 2, 3. Not bad, huh? Not bad. So, it helps that they're all connected to the same brain. Okay, right now you have 4 independent brains doing this. Okay, let me show you. So this was a walk. Let me show you a trot. I can't actually see the cursor. I don't know if it's playing. Unfortunate. Let's just go to the next. Beautiful trot. What's happening? Can you see?
Yeah.
What's happening?
First and third, second and fourth.
First and third, but at the same time. Second and fourth at the same time. So it's a diagonal gait, but the timing is different. Can you think what the brain and spinal cord have to do when a horse is doing the walk, 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 and suddenly decides it's got a trot? It suddenly changes the timing, right? It's the same action, but the timing is different. And then, here is a learned gait. Okay, the trot and the walk are natural. Here is a learned gait. This is pacing. I just got this from the web, okay? I know nothing of horses. How is pacing different from trotting?
The legs.
It's still 2 legs at a time, but which 2?
Side.
It's a lateral gait, no? It's not diagonal. Okay, so this tells you how many different things a horse does, okay? And I'm going to do a last audience activity. I'm going to divide you up and make you the legs of a horse. Okay, why? When we made these 4 people clap and move, we were just representing the sound, right? That's what you hear. But think what's happening when a leg has to move so that the hoof hits the ground. Okay? Everybody flex your biceps. How many signals have to go where? Biceps have to flex, right? What extended? The opposite one extended, no? Triceps. So and that's just here. So when a horse does this, you can imagine how many signals go to how many muscles in a proper sequence so that the hoof sounds exactly when it's supposed to. Okay, and the hoof sound is just a readout of the gait. So now I'm going to divide the audience. Let's see. Let's have this natural down the middle here. And if I can have you guys sort of count yourselves on this side, the back row. You don't have to move, just think of yourselves in the back. And then I'll have the first 2 rows be 1. Okay, and all of you, including you in the back, be 3. Here the first 2 rows are 2. And all of you are 4. Okay, and now we're going to clap. So we're going to first walk. 1, 4, 2, 3. 1, 4, 2, 3. 1, 4, 2, 3. 1, 4, 2, 3. 1, 4, 2, 3. 1, 4, 2, 3. Okay, now trot. Ready? Trotting is diagonal. Okay? So I'm just going to go clip, clop, clip, clop. Ready? Go. Clip, clop, clip, clop, clip, clop, clip, clop. Okay, now gallop.
1 and 3 and 2.
Oh, okay, that's pace. Come on, pace. Ready? Go. Gallop is different. Gallop is, tabadik, 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 tabadik. But actually there are 4 sounds in there. It goes tabadidik, tabadidik, tabadidik. And I don't know how to do it. But that's homework. Okay. So, we're close to the end of what I hope was an evening where we share some of the magic of what we do. I hope we've raised questions about everything you think is real. And how marvelous is it that all of it comes from this mushy blob inside our heads. Okay, a mushy blob so uninspiring to ancient Egyptians that when they mummified bodies and they preserved all the important organs, they first drained out the brain and the mush through the nose. Okay, they thought it was mucus. Okay, this mushy blob creates the world we see. Okay, it represents it in our head, all of its richness and all of the imagination and creativity we derive from it. All of our dreams come from this fantastic structure in our heads. And this structure, I claim, tells you what the purpose of an egg is. Any guesses? What is the purpose of an egg? Do you know what I'm going to say? To shield the bird forming inside. Yep, that's the purpose of the egg shell. The skull of the egg. What's the purpose of an egg? To make another egg. Exactly. Okay, the purpose of an egg in terms of biology is to make more eggs. Otherwise the species is dead. And the most humbling insight I've learned as a biologist is that life and our hopes and dreams and dramas and oh he doesn't love me and poetry and creativity and books and imagination, everything is just a little bits of milestones along the way from one egg making more eggs. So even as we marvel, let's not take it all too seriously. It's all Maya, it's all a matrix. We're all feeling our way. But as long as it's fun, let's enjoy ourselves. Thank you very much.
Will there be any more questions for Shubha? Would it be okay to answer the question? No.
I have got, when is your next lecture at
When and where is my next lecture? St. Xavier's College. I'm invited to be Professor X in their series called Paradigm in January, 1 month from today. Don't ask me what Professor X is.
Xavier? We'll explore it then.
Maybe Professor Xavier's, yes. But
What is the time and the place? Time?
I don't know yet. I think midday, sometime midday on the 18th of January.
Repeat the name of the book you said,
Phantoms in the Brain
Phantom.
by V.S. Ramachandran.
When was it?
When it was written,
Recently or very old?
I bought a copy 15 years ago. But if you Google V.S. Ramachandran, he's a lovely writer. So actually, if you want to read about the brain in sheer poetry, okay, you should read Oliver Sacks, The Man Who Mistook His Wife for a Hat. Okay, this is an old book written in sort of old language. He was a neurologist who described each case study in just poetic ways. This is a person whose brain is executing it this way and that way. And so, we understand how the brain might be constructed and how it might work by looking at what happens when something goes wrong. So, you can remember old memories, but you can't make new ones. Therefore, the storage site of memory may be different from the recording site. So, by the mistakes that can and cannot happen, you extract what might be. And that's what we continue to do in the lab. Okay, we make mutations in brains of mice, and we look at what can and cannot form. So we have mutations where the signaling center, the lighthouse, becomes huge, such that you have everything being signaling center and nobody for it to respond to. Okay? Or you have it where it's in little bits and pieces all over the place, and then wherever it is, it forms the memory center next door, because that's what the brain is supposed to do. So we explore this by testing what happens when things go wrong. And the broad hope then down the road is once we have the framework of how the brain normally develops, then we may have an insight into these heartbreaking disorders that children are born with, or that come to pass.
There are very interesting TED Talks as well, right? Mr. Ramchandran as well, done by all of this.
Yes.
See, I just had a very, I don't know if this is more in psychology, addictions and habits. Is there a discussion or some techniques that you know experience?
Addiction.
How to break an addiction or based on the brain, is there some
Even certain traits, kleptomaniacs, you say, it's a trait. I mean, it is there.
So the description of normal human behavior is quite wide. And I think our goal should be to make it wider still. Okay, I mean, you may know what I'm alluding to. We, in the past and maybe even in the present in pockets, we have extremely limited notions of what normal is and anything outside. I mean, it used to be, if a bride from a neighboring village was, not normal because she didn't, my mother used to be criticized for not making chapatis in a certain way. The definition of normal is very, very limited. And now we are getting broader and broader in our definition of normal. We have many practices, behaviors, abilities that were thought not to be normal before, but now are getting included in this umbrella of normal, and I like that. I think we should have an extremely broad stretch of what is normal, because it's all a manifestation of a brain and a mind, and as long as it doesn't impinge on somebody else, we should include it in our definition of normal. So this said, we can talk differentially about kleptomania and addiction, right? I mean, if somebody collects things, I'm not fussed about it. If they are addicted to something, that's sort of using the brain's pathways in a way that then leads your own life out of control. So addiction sort of works by a continuous internal reinforcement of signals in the brain that should have damped. Where a brain cell throws signals on the next cell, that signal is supposed to be mopped up and damped so that the next cell is fresh for the new signal. But drugs of abuse keep that signal around longer, so the next cell is feeling the buzz for longer and it feels good. And when it feels good, you do it again and again and again. Then that sort of leads into a spiral. So that's a disorder. I think you may have read more than I'm saying about this. So what would you wish me to address? I'm not even sure I'm the expert.
I have one question. Talking about this creativity and innovation. Can the brain think always out of box? Or what are the conditions which need brain to think out of box? Can it be cultivated?
Can out-of-box thinking be cultivated? I would say absolutely. And, by inviting people like me who could have sat in our labs doing our in-box thinking to come and do sessions like this. So this game was actually originally invented for Science Day for a group of 7th standard students. And it has now gone international. I've presented it at workshops, international workshop, at 3rd standard things, at sessions like this. So this takes us out of the box. It makes us stretch out. So the little skit that we did, we put it together in 2 days and we tweaked it and shaped it to be scientifically accurate. Everything I said about it was scientifically accurate. Okay, the jacket represented something, the fact that other students didn't have, kept the jacket on meant something, the different colors meant something. So it's a way of stretching us out of box. You folks coming here, you've walked out of boxes to come here. I don't know what backgrounds you have, but it's fantastic, right? And the questions you're asking, this interaction is taking us out of box. So there's a popular saying, use it or lose it. So making your brain do things that it's not used to doing keeps it stimulated and makes you broader and more creative. So broadly that would be it. Give as many new experiences as you can to the brain. Sorry.
Right side of the brain, left side of the brain.
Yes.
What is the difference?
What is the difference between right and left side?
And does it work together?
Okay. Now, the two halves of our body will look similar, mirror image from the outside. For evolutionary reasons associated with what makes things as beautiful. So if you have an asymmetry that's not regarded as, so there was a study done where people averaged many faces and showed a composite image of many faces averaged and asked people of the opposite gender to line them up and say which is more beautiful than the other. And the more faces you average, the more beautiful the person was thought to be. Okay, and the more you average, the more you get absolute similar, bilaterally symmetric looks. Okay, the reason I'm saying this is that everything inside is not bilaterally symmetric. So we should not be amazed, oh my goodness, the left and right brains are not identical. It's only the outside that's identical. The two lungs are not identical. The heart's off to one side. The digestive system is this way and that. This very little, even the kidneys are one up and one down. Okay, so almost everything is asymmetric, and yes, the brain is also asymmetric. Different abilities are processed more or less in different halves, and the two halves do talk to each other. There are some fantastic split brain studies, okay, where Roger Sperry is the scientist whose work you want to look at. Looked at patients and then animals where the connections between the two halves of the brain are severed, and they're severed not for fun, but because patients had seizures and this prevents a seizure from transmitting. And then if the connections are severed, you can look through boxes and present different information to different halves of the brain. One half of the brain can process language but can't speak it, one half of the brain, So you can do these tests, and they allow you to test what two halves do. I mean, I hope that's a broad answer to what is interesting, and then you can look up more. I'm sorry, there was another question. Let's go ahead.
There's a lot of talk these days about mindfulness and how mindfulness can impact your intuition.
About mindfulness and intuition. I don't know what mindfulness is.
Mindfulness, I can define it.
Observing the mind, being more aware.
More aware of what you do.
Self-aware, I suppose, right? I would agree.
Okay, I was just curious how your brain, how it affects, how being mindful is supposed to affect your intuition, your intellectual activity, your creativity. How does that look?
So I can't answer how or why. I would just say I'm not surprised. Because it's sort of linked to your out-of-box question. If you continue to take, so your brain is a highly connected processing system that's looking for sensations, right? Think about it, it's still in this little skull, and all it gets is what the eyes care to look at and what you care to touch. Okay, so in fact, people have done experiments on sensory deprivation. Okay, you can, for short periods of time, with all the ethical clearances and consent, ask people to volunteer for studies where they go into sensory deprivation. They live in a dark room in a suit that's so soft that you don't actually feel anything, and there's no sound and so on. You can do sensory deprivation studies. Okay, to ask what happens. So our brain likes sensation, our brain likes stimulation. Okay. The reason it's good to wear a hearing aid if you're hard of hearing is that your cortex does not like being silent, and if you expose it to being silent for a long, long time, it's not going to be happy processing even the small sound that it hears. So giving it stimulation and input is likely to be good for it, and mindfulness seems to be more of that. So how can it be wrong? How it actually helps and how it helps make you more intuitive, it's a complex question. It's a complex question. So it's like saying, if you're learning Bharatanatyam, going and watching 8 other different dance forms and maybe doing workshops in them is going to make your Bharatanatyam more interesting. I mean, you would know it as well.
Just one last.
Sorry. Yes? Okay, so can we just answer the hands I've seen? There's a lady in the back there, and I saw your hand, and then we can do it as quickly as possible. Yes?
Essentially, medicines or supplements that are trying to say that they will enhance your memory are fooling us.
Medicines that enhance your memory
They
are
claim
fooling
that-
us. I'm sure there are drugs that make your circuits perform at a higher level. They would probably slide you down the drugs of abuse path. Okay, so yes, you can make your circuits perform at an enhanced level. So improving memory might just be an enhanced recollection. I would caution greatly against that. So
You can stimulate, you can't enhance something.
semantics.
I know. I'm a teacher of semantics.
Yes.
As far as memories, you must be having some idea where it might be. You may not be sure.
You may have some idea where it might be. There is no one bit of the brain which if you take out, you'll suddenly lose all your memories. Okay? But you do lose your memories when you have senile dementia, Alzheimer's, where basically all of the cortex begins to slowly corrode. So the best I know is it's still distributed.
You don't have, besides the hippocampus, you don't have any idea.
Hippocampus is the recorder.
Yes,
It's only the recorder.
yes. And emotional also, it's emotional involved.
It's the amygdala kicks in for emotional memories.
Yeah. But you don't have any other place where you could think it might
Think of the challenges ahead of us.
Yes.
Send your children to do research with us.
Yes.
Yes, last question.
I have two young kids. Just want to, does our education system optimizes what we know about the brain and what we know so far?
That's our education
Is there
system.
something we can do with them so that we enhance? Because it's a lot more correct information to somebody.
Okay, I will give you two opposite answers. Okay, one, there are certainly modern ways of educating children. People say children these days are smarter than children before. Okay, that's because children before, long ago, were not barely sent to school until the age of 6 or 7, right? Now you have them in school at the age of 2.5, and you find those, little minds perform by all benchmarks. Okay, yet, I would say, some of the greatest insights of known to humankind and some of the fantastic creativity came from people who haven't gone to school and came from those ages when the education system was poor.
True.
Okay, I went to a very regular school, St. Joseph's School, a regular school. SSC, it was the nearest school. We went there, we came back, we played. The most creative thing in my life was my mom. Okay, and I looked forward to summers. Okay, and here I stand today, a scientist at TIFR, doing ribbon game with all of you. Would I have done it differently or better or worse if I had gone to one of those enhanced schools? God only knows.
Yeah.
Amazing, thank you.